Good afternoon, good morning, good evening, whenever you're looking at this. So just to start the video, a bit of revision, pause the video, have a look at this picture. What's this diagram showing? What's it illustrating? Okay, so can you see what it's demonstrating here? So we can see that we've got the same neurotransmitter in both diagrams. We've got acetylcholine here, we've got acetylcholine here. But we're on the left-hand side with the skeletal muscle, we can see that acetylcholine, the receptors are different here for the skeletal muscle than they are here for the heart muscle. So the effect is different. Okay, so it promotes muscle contraction here in the skeletal muscle, whereas it inhibits muscle contraction here within the heart muscle. So that's down to the type of receptor that is present and not the neurotransmitter itself. Okay, so that's a key point that we learned previously. So let's have a look at these 10 questions. The first 5 are problem solving, and then the last 5 are from a topic that we've just done recently. Have a go, and the answers will come up in 5 seconds. Okay, so there are your answers. Mark your answers, and if you need help, get in touch. You know where we are. So we're going to look today specifically at neurotransmitters. So we've talked about neurotransmitters. We've mentioned neurotransmitters already. We're just going to look at them in a little bit more detail today and what they do. So here are the key points. Pause the video and have a read through the key learning points for this lesson. So first of all, why is it so important that neurotransmitters are removed from the synaptic cleft once threshold has been reached? What would happen if the neurotransmitters weren't removed? Have a think about it. So, what would be the problem? Well, if you imagine that we need 4 neurotransmitters to travel across the synapse and bind to the receptors on the other side in order to reach threshold. So an electrical impulse travels through this axon. It stimulates the release of thousands and thousands of neurotransmitters into the synaptic cleft, and they start to diffuse across the synaptic cleft. Threshold is reached, and as soon as you reach threshold, the message that you are sending, the signal that you are sending, has been passed on. The electrical impulse in the neuron, or in this case, a muscle, will be passed on, and it will cause the muscle to contract because threshold has been reached. Potentially, what could be the problem if these neurotransmitters are not removed from the synaptic cleft? Well, potentially, you could end up with something called continuous stimulation. So you want to contract the muscle. So you need to be able to reach threshold for that impulse to be passed across. But once these neurotransmitters come out of the receptor, if the cleft was still full of neurotransmitter, potentially more neurotransmitters could join to the receptors, and potentially you could reach threshold again. So potentially you could send another signal. You could cause the muscle to contract again, even though you don't want it to contract again. You just want it to contract once, and then you want it to stop. So it's important that very, very quickly, once threshold has been reached, that these neurotransmitters that are in the synaptic cleft are taken out of the synaptic cleft. And that's basically what this slide's talking about, the prevention of continuous stimulation of either muscles or the postsynaptic neurons. So how are the neurotransmitters removed from the synaptic cleft? Well, there are 2 main ways. The first way is the simple way, and that's just a simple a reuptake of the neurotransmitter. So if you can see here, here's the presynaptic neuron, And there's the post-synaptic neuron with the receptors. And you can see the vesicles containing the neurotransmitters, and they're being released into the synaptic cleft. They move across, bind to the receptor, threshold is reached, and the impulse is passed on. Now, a lot of these neurotransmitters will then be retaken back into the presynaptic neuron through a protein molecule in the membrane. So they'll be actively transported back into the presynaptic neuron, into the axon, and then stored again in vesicles, ready for the next time that an electrical impulse travels and stimulates them to move to the membrane again. So an example of a neurotransmitter that is just simply reabsorbed through a protein is noradrenaline. Another way that neurotransmitters get taken back into the presynaptic neuron is by enzyme degradation. Now, the example here is an example of acetylcholine. So acetylcholine is the neurotransmitter. And you can see here in the synaptic cleft that we've got lots and lots of acetylcholine. And then on the membrane here, what happens is that acetylcholine, the neurotransmitter, gets broken down by an enzyme. And in this particular example, the enzyme is called acetylcholine esterase. Acetylcholine esterase. So it breaks down the acetylcholine into acetate and choline. And then if you look over here, you can see that those products, the choline and the acetate, the things that have been broken down, are then able to move back into the presynaptic neuron. So they need to be broken down by an enzyme or degraded by an enzyme into a smaller product before they're reabsorbed into the presynaptic neuron. Once they're in the presynaptic neuron, those two molecules get built back up again into acetylcholine, which is our neurotransmitter, which can be stored inside the vesicle, ready for use again. Now, that process requires a lot of energy, and we mentioned it in the last video as well. And that's why, generally speaking, you will have an awful lot of mitochondria present at the end of an axon, because energy is needed for the reabsorption of these neurotransmitters. So the key point that you really need to take away from this little section of the video is, why is it so important that neurotransmitters are removed from the synaptic cleft? Okay, have you got that? So that's to prevent continuous stimulation. And then the two methods, reuptake through a protein in the membrane for neurotransmitters like noradrenaline, and also enzyme degradation, the breakdown of the neurotransmitter by an enzyme. The products, the smaller products, are then taken back into the presynaptic neuron and built up into the neurotransmitter. We're now going to move on to a slightly different section, which is about how neurotransmitters, different neurotransmitters, affect your mood and affect your behavior. So the two that we're going to look at are dopamine and endorphins. And you really need to know the effect of these neurotransmitters, and also when these neurotransmitters are released. So we'll start by looking at endorphins. So normally in your body, when you feel pain, that pain is a message really that's being sent through your nervous system and through your nerves. So if you take a look here, you can see there is a neurotransmitter here, and if you are feeling pain, that neurotransmitter is released across the synaptic cleft. It binds with the receptor, you reach threshold, and that message of pain travels on through the next neuron. So what endorphins do, they're a natural painkiller in effect. And what they do is they bind to receptors, and they basically block the release of the pain neurotransmitters, if you like. So for the pain message to be passed on, those neurotransmitters need to be released. But these endorphins here, by binding to these receptor sites here, they basically block the release of these neurotransmitters. And so in effect, it stops the message, the pain message, moving from one neuron to the next neuron. So in effect, it filters out the pain message, or the pain impulse, if you like. When are endorphins released? Well, they're released in larger numbers in these particular situations. So severe injury, prolonged continuous exercise, physical and emotional stress during sex, and when you eat chocolate. Okay, so endorphins tend to be released more during these activities. The other neurotransmitter that we need to know about is dopamine. And dopamine is a neurotransmitter that really makes us feel good, makes us feel happy, gives us pleasure. And it's linked to parts of the brain that are known as the reward pathway. Parts of the brain that are known as the reward pathway are just neurons that release and respond to this neurotransmitter, dopamine. And we release dopamine as a neurotransmitter, and it makes us feel good when we behave in a way that is useful to us or beneficial to us. So for example, the example that's given is when we're really hungry and we eat after we've been really, really hungry, we can feel really happy. We can feel real pleasure from eating food. And obviously the act of eating food is beneficial for us in terms of our survival. So in effect, the reward pathway, it encourages us to carry out behaviors that are beneficial for us to increase our chances of surviving. If eating food wasn't a pleasant experience, if we didn't enjoy it, then obviously we would do it less, or if at all, and potentially that could be dangerous and quite serious. It's the same with sex. If sex was a really unpleasant experience, then obviously the human race would be having problems, wouldn't it? Because a population relies on reproduction in terms of it being successful. So these sorts of behaviors are rewarded with feelings of pleasure because they are beneficial, not just to the individual, but also to the population. So we're going to have a look at a couple of disorders that are related to neurotransmitters. You'll have heard of a few of them already. So depression, Alzheimer's, and Parkinson's disease. You don't need to know great detail about these disorders at all. And we're just going to look at we're going to look at three main drugs. We're going to look at agonists and antagonists. We're also going to look at inhibitors as well, and how these three types of drugs are used to treat various disorders that are related to neurotransmitters. So first of all, we're going to have a look at agonists and antagonists. You need to know the difference between an agonist and an antagonist. So on the left, we've got an agonist. So an agonist is a drug that is very similar to the neurotransmitter. So it in effect mimics the neurotransmitter at the synapse. So that you can see here, the drug is able to bind to the receptor on the postsynaptic neuron. And when it binds to the receptor, it makes it easier to reach threshold, and it activates the cell, and it passes the signal on. So that's one way a drug can work. It can act as an agonist, and it can stimulate the neurons. Antagonists are opposite to that, really. So you can see the antagonist drug here, it is able to fit in to the receptor, but it only fits in to the receptor and blocks the receptor. So it basically it stops the receptor from receiving any neurotransmitter, but it doesn't activate the cell itself, unlike the agonist. Okay, so the antagonist blocks up the receptor and blocks the action of any neurotransmitter with that particular receptor. A third type of drug are drugs that inhibit the enzymes that degrade the neurotransmitters. So do you remember we said that for a neurotransmitter like acetylcholine, for it to move back into the presynaptic neuron, it first of all needs to be broken down by an enzyme so that the products can move back into the neuron, ready for when it's needed next time. So we're going to show you basically how an inhibitor, a drug inhibitor, works. So this is used for treating Alzheimer's disease. So with Alzheimer's disease, there is an issue with the amount of acetylcholine that is released. So somebody with Alzheimer's don't release a high enough concentration really of neurotransmitter. And this neurotransmitter in this particular part of the brain is linked to memory and to learning. So how can a drug help to treat Alzheimer's disease? Well, we said, didn't we, that normally what happens is the acetylcholine gets broken down into choline and acetate and then gets put back in out of the synaptic cleft back into the presynaptic neuron. And the breakdown of acetylcholine in the synaptic cleft is carried out by an enzyme called acetylcholinesterase. Well, if you can inhibit the activity of that enzyme, then you stop the breakdown of acetylcholine in the synaptic cleft. And so in effect, what you do is it leads to a buildup within the synaptic cleft of acetylcholine, which then in turn increases the chances of threshold being reached at that particular synapse. So we can see that happening here. Okay, so there's the acetylcholine being released from the presynaptic neuron. It travels across to the receptor, and then normally it would be broken down and it would move back into the presynaptic neuron. But what we're saying here is that there is an inhibitor. The drug inhibits the enzyme that breaks down the acetylcholine, and that allows the acetylcholine to build up in the synaptic cleft. Another disease that is affected by neurotransmitters is Parkinson's disease. So you can see the difference between normal and somebody with Parkinson's. There is a shortage, there is a lack of a particular neurotransmitter called dopamine. So obviously somebody with Parkinson's is going to struggle to reach threshold with that number of neurotransmitters being released. And with Parkinson's disease, it's a part of the brain that's affected that's responsible for planning and controlling body movement. So one of the side effects of Parkinson's is they find it difficult to control the movement of the body as the Parkinson's develops, because it's a degenerative disease. It gets progressively worse. Again, for treatment of depression, there's various ways of treating depression, but one way is to, again, stop the uptake of the neurotransmitter. So the neurotransmitter here in a healthy person would release serotonin, and that would travel across the synapse. And then normally it would be reabsorbed, taken back up through a protein in the membrane. But again, if you can block that protein, if you use drugs to block the protein, block the pathway back into the presynaptic neuron, well, that increases the concentration of the neurotransmitter in the synaptic cleft, and again increases the chances of you reaching threshold, increases the chances of the signal being passed on in the next neuron. The final bit that we're going to look at is recreational drugs. So there's obviously legal and illegal drugs. I'm needing my caffeine just now, but there's other drugs that are legal that people take, cigarettes, alcohol, and then obviously there's a lot of illegal drugs that are available. But all these drugs, whether legal or illegal, they affect neurotransmitters within your brain. They affect the chemicals in your brain, and specifically they affect this area called the reward pathway. So a lot of these drugs do give you a feeling of pleasure. That's why people take them, because it does affect the reward pathway in the brain. But it can also affect other parts of the person. So we've listed 4 areas here. So mood. So for example, alcohol is actually a depressant. So over a period of time, it can affect your mood in a negative way. Cognition. So in other words, your ability to solve problems and your ability to think are affected by drugs. Your perception, what you interpret, is affected by drugs, and your behavior as well can be affected by drugs. So depending on the type of drug that it is that you're using, well that will dictate the sort of behavior that it brings out in you. So how do drugs work? How do recreational drugs work? Well, really, it's just a recap of what we've learned already about neurotransmitters. So number one, they stimulate the release of natural neurotransmitters. So some drugs will just stimulate more neurotransmitters to be released into the synaptic cleft and increase the chances of reaching threshold. Some of them act like neurotransmitters. So we talked about agonists. So they bind to the receptors and they bring about a response. Some of them act like antagonists. So these will block the receptors and reduce the chances of reaching threshold. Some of them inhibit the reuptake of the neurotransmitter. So by blocking the protein that usually allows the neurotransmitter back into the presynaptic neuron. And then finally, some of them inhibit the breakdown of the neurotransmitter by enzyme degradation, which we've talked about. So that's how recreational drugs work in one of those 5 ways. The final bit that we're going to look at is drug addiction. So there's a definition there of what drug addiction is. But we need to understand 2 forms, in effect, of drug addiction. One is caused by antagonists, and that's called desensitization. And one's caused by agonists, and that's called desensitization. So let's just remind ourselves of the difference between agonist and antagonist. So this is normal, this is natural on the left. Before the drug, you've got a natural neurotransmitter that binds to the receptor, you reach threshold, and the signal's passed on, you get cell activity. Agonist drugs are basically very similar to the natural neurotransmitter. They bind to the receptor, and they increase the chances of reaching threshold because you have a higher concentration within the synaptic cleft, and so you have enhanced cellular activity. And then the antagonist basically blocks the receptor, stops the natural neurotransmitter from binding, reduces the chances of reaching threshold, and so reduces cell activity. We haven't got time to go into all the different types of drugs, but you could maybe do a little bit of research into forms of recreational drugs that are agonists and that are antagonists. So what's the result of these different forms of drugs? Well, antagonists drugs, basically what happens within the nervous system is that the nervous system will compensate for reduced stimulation. So it would normally have greater stimulation, but once the drug is in the system and blocking the receptors, the nervous system adapts itself. And so the postsynaptic neurons, they will basically develop more receptors. They'll increase the number of receptors on the postsynaptic neuron. So an increase in the number and sensitivity of those receptors on the postsynaptic neuron leads to this form of addiction, where the person craves more of the drug. With desensitization, this is what happens with a drug that's an agonist. So not antagonist, an agonist. So because you've got more of this cell activity, more opportunity to reach threshold, again, the nervous system adapts and it changes. And this time it reduces the number of receptors, and the sensitivity of the receptors reduces as well. And what you find is, because there are fewer receptors and they're less sensitive, that the person never gets the same effect again, even though they keep taking the drug at the same dosage. Because the nervous system has changed, they never get the feeling that they once had. And this is called drug tolerance. So in order to get the same feeling, they need to take a larger dose of the drug to bring about the original effect. So go back, have a look at the two types of drug addiction, one caused by drugs that are agonists and one caused by drugs that are antagonists, and just run through the difference between sensitization and how that adapts the nervous system with more receptors being on the postsynaptic neuron, and they become more sensitive. And with desensitization, the nervous system adapts again, but this time the receptors are reduced and they become less sensitive. And so you need to take more of the drug to get the same effect. Okay, so have a look through the key learning points, think what your questions are, and use these statements to come up with your questions.